organizer kindly invited me to give a talk about alternative splicing, basically about visualization. And my immediate response was no, of course, because I don't have the time and it's not my field. But luckily, I did not press the send button right away, and I thought about it a bit more and realized I always say that's so important to get progress in this field, so I better put my time where my mouth is, basically. So I'm very happy to be here, and I'll give a gentle introduction about alternative splicing with an emphasis about visualization issues. Okay, so let's start. Central dogma for information processing, DNA transcription into RNA, and I know there's people with different backgrounds in the audience, so we'll go through that. And then translated, in many cases, into a protein. And what people are less familiar with is that we have, if we zoom in into the RNA stage, then we have actually a pre-messenger RNA, and we have elements in it that are called exons that are spliced together. The other elements, introns, are not shown here, are removed. That produces the messenger RNA, simplifying the process, of course, and then that is translated into protein. And that brings us to the first visualization, is how do I visualize a transcript from the DNA? And this, we've seen it time and time again. People sort of assume everybody knows about it, but that's somewhere it became a standard. So these are, again, the exons that we saw before, in between introns, or whatever the DNA sequence is, in between. And then what it's implicit here is the stop and the start codon are shown here in red. And the smaller rectangles are the 5' untranslated regions UDRs and 3' UDRs. And then basically all the big rectangles are the actual coding sequence that are spliced together and translated to protein later on. OK, so that's visualization number one. Back to the central dogma. The interest in my field is that we can actually get something that's called alternative splicing. So if you just missed it, the red exon, in this case, was removed, so it's not included, given rise to a different isoform and a different messenger RNA, and then translated to a different protein that might have a completely different function, or a slightly different function. And then, of course, we need to visualize too, so we can do that too, right? That's quite simple. OK, so alternative splicing significantly increases complexity. So if we go across the species, we go all the way to human, we can see the vast majority of the human genes have multiple exons, and a vast majority of those have alternative splicing. And if we check it, so most of the alternative splicing that we detect is tissue-specific, indicating functionality. And there's estimates for a large fraction of complex human disease mutations that affect alternative splicing. Just as one example, this is the first one I know of, at least with functional relation to this. This is from '93, a very famous example for the BCL2. And you have here two isoforms. So basically, this axon has two splice variants, either using this end over here or this end. And the longer version, the L version of it, is related to anti-apoptosis effect associated with different types of cancer. And then the short one, even the introduction of it, causes apoptosis in many different cancers. So that's just one example. OK. Of course, there are different. So this is one type of splicing changes. There are many different ones. Where are the sources for our knowledge about alternative splicing? So splicing in general was identified back in the 70s. But a lot of the information originally came from EST and cDNA libraries that were built along the years. And then, as John reviewed, along came the microarray years with an explosion of data. And that gave us another indication for possible splice variants. They came from either axon tile arrays or microarrays that were designed with special junctions for probes for junctions across exons. And then what we have now is the explosion of data that comes from high throughput sequencing. So what types of alternative splicing can we get from this data? This is one example. This is one of the first papers, Wang from Burge group here at MIT, end of 2008. several different human tissues, and this is an example of the type of splicing that we see. Here we see, this is the map, so constitutive exons here are black. The alternative one is the white one, and we have short sequence reads that were mapped, so body reads are mapped directly to the exon, and then these with the dashed lines map across, so these are basically junction reads. So they map across junctions, and then the red ones are junctions that support the inclusion of the isoform, and then the blue ones, according to their notation, support the exclusion. So this is a skipped exon. Either it's included or excluded, and there are many other different types that they mapped. Retention of an intron, alternative 5' 3', mutually exclusive, alternative first, alternative and tandem 3' UTR. And overall, just to show the complexity, this was a limited depth of sequencing and only a handful of tissues, still over 100,000 splicing events that they mapped. With this being the most common, cassette exon, we'll get back to it later. Okay, so what this data gives us is very local information of where exons are, which of the exons are connected together, and one way to put it together, this information in terms of visualization, is a splicing graph where we can see which of the exons are connected together, okay? But, so it's a compact representation in terms of the visualization, and it's more than just the list of exons, right? Because we can see which go to which, and it may represent the only knowledge that we actually have. For example, from short reads from HTS data. I should point out that there are longer reads that are coming from, for example, PacBio that is coming up with, say, 1,000 bases. So that would cover maybe several exons, and that would introduce sort of constraints onto this graph, which would be interesting to visualize. But anyway, my point is that it's missing a lot of information because isoforms are underspecified here, right? The number of passes here along these graphs is exponential with the connectivity. And then, for example, where is the start, where are the ends, and which combinations actually exist, okay? And just to illustrate that this is a complexity that is building up more and more. So it's just illustration for one possible gene, but just to see the complexity in terms of the visualization that we need to handle and then the types of problems that are coming in. So this is a quote from a very recent paper from Pritchard's lab at Chicago. And they used 75 individuals. People mentioned here about variations between individuals. Only a single tissue. And they detected 150,000 new splice junctions. A lot of them are low abundance isoforms that are probably noise in the splicing, but not all of them. And which, right? So when you start visualize all this, with these types of visualization, you need some way to indicate where the data come from. So kind of questions that comes into mind are, what are the actual transcripts that come out of this, right? How much support each splice variant actually has, right? If it's low abundance, is it noise, not noise, et cetera, et cetera. And in what context? What tissue? Is it what disease, et cetera? Okay? And let's look at a real example. So it looks like this. Right, so this is RBM39, and this is a genome browser, everyone's favorite, as they mentioned before. And where's the problem, right? So I think to sort of try and start mapping the problem is, what do we use visualization for, right? If I just want to show you that it's complex, I think I convinced you right there. But we want to use visualization to understand a concept which is harder to understand, or we want to use it to explore new things that we don't know before. And I would argue that it's not necessarily as easy. Some of the things, because as it was smartly built in a way that we can have different tracks and conservation, et cetera, that was mentioned before, you can see different relations and you can come up with different hypotheses, but it's really, really hard to go back and forth and say, study this specific exon and splice variants of it, and where it shows, and what tissues, et cetera, right? It's not built for that. Okay, and just to show that the UCSC browser is not alone, this is an Argo. That's a tool built here by the Broad who hosts us. So of course I took to show their tool, and this is a standalone application, a Java-based. And then we can, again, you'll always have the sequences here, and this is where you probe the sequence DNA or messenger RNA or whatever, and this you can do finding with this panel over here. And they have the integrated genomics viewer, which John mentioned in the talk before me. And that's the same gene, by the way, okay, RBM39. And again, you can see the complexity. And here you can have different data sets and different attributes, as they call it. And you can map them and see the relation. And again, you can see here the chromosome. And very nicely, you can zoom in and out. Because it's a standalone applet, you can just go in and out very quickly, which is nice. But still, I mean, what are we missing in terms of visualization of alternative splicing? So I think a very, to summarize sort of what's the lesson. A very known principle that we all heard is this WYSIWYG, right? What you see is what you get. So we use it all the time. And I think so the underlying implicit principle that we're suffering from here is something like this, which you can try to read as YAKWID, but I'm not sure if you actually want to say that. But basically what I'm trying to say is you can't see what you don't get, right? And what do I mean by that? So I went through those tools, and I found this interesting quote from the Argo manual. It says, the data displayed features on sequence. Simply put, a sequence is a string. A feature is a location on a string. Features are divided into logical and display groups called tracks, okay? So what I'm trying to argue here, and I think you could almost hear a hint of frustration from some of the questions that we heard through the day, is everything is this string as the base of all our visualization, and everything is built on top of it or as flat files that introduce attributes with relation to it. And what I would argue is that apparently, at least for someone like me who's studying splicing, but I'm guessing for many others, we want to have a relational paradigm. So different entities are represented, and the different connections between. There might be genes, there might be axons, there might be introns, and there are different relations between them. And then the relational information from them, okay? And representation for isoform-specific data, which is currently missing. For example, coding, non-coding, PTC, whereas PTC is pretermination codon introduction, validated transcripts end and starts at different levels. And then I told you, for example, high throughput data and many junctions that are mapped at different levels. And so how much support do you have and how sure you are? So you want to have sort of a control knob over that. Canonical splice junctions like these, many information that is specifically related to isoforms. But you, type of experimental support, where the data came from, is a cDNA, EST, if that is so, from what tissue, et cetera, et cetera. I would like to be able to see all that and the level of the experimental support that I mentioned, and context information, as things like I mentioned before. And I will show you one example which is more geared towards isoforms, I think, which is already out there. Maybe not the only one, but sort of in the direction. So this is the ACE view by NCBI. Okay, it's a rather old tool that has been there, and it's based on EST cDNA libraries. So this is how they map their different isoforms. And now you can see a lot of information that they use visual clues for. So they label them like this. If it has an underlying here, then it means a different level of supports. These are whether they're actually detected the start and the end and what level of it. And color means that they're the same. And the height of the color means the level of support in terms of sequences in their databases, et cetera, et cetera. So it's really geared towards that. And if you click anywhere here, it gives you more information about the protein structure, frame shifts, the supporting sequences, et cetera. So it's more geared towards that. That's a different view that they have. So another thing that I'm sort of pushing towards to, in a sense, in that talk is it's more, it's not, it's Flash-based, okay, or animated GIF, so it's more dynamic. And to summarize sort of the, and of course, now what happens is you get this, say, in ACE view, but you're missing all the other information, the great information that the tools that I just showed you had in them in terms of bringing out all the other information, all these attributes, all these mappings, and crisscrossing all that information that it can actually come up with a valid hypothesis, right? So, just to summarize, what I'd like to have is appropriate filters or control knobs, whether discrete or real. So real was, say, the level of support that I'm getting from high throughput sequencing, that this splice junction is actually real, yes or no. And discrete might be different tissue types, disease versus normal, subtypes of disease, et cetera, right? And alternative representation for transcript or data entities. So I can switch between them seamlessly, right? If I go all the way in, maybe I switch on the sequence to maybe its putative secondary structure or where it has free regions, et cetera, et cetera. Okay, and I can do that as I zoom in and I zoom out, and I can switch between different representation of the same data according to what I actually need right now or according to the zoom level. So it's sort of in there when people are talking about the zoom level, but I think it's more complex than that. Okay, so switch the entity focus, say from the, a region in the chromosome to a gene, and then maybe I want to concentrate on that specific exon, and then I want, when I'm focused on it, maybe I want to see what are the transcripts that it belongs to and where was it implicated, and the SNPs and the structural variations that are related to it. So now the exon becomes my focus of interest, right? We need a matching rendering engine for all that nice ideas, and a relational database that, as I understand, and I don't know the details, is implemented in various ways already to make all these queries, etc., efficient. So it's more maybe about the visualization engine and the sort of conceptual thinking of it. Right, and then a flexible interface definition. With respect to that, people who maybe come from object-oriented programming, object-oriented design, and graphical interface would recognize it as a design pattern for visual proxy. So just in a nutshell, what I mean by that is separation between the data and abstraction level, and then a visualization of it that you can switch. So you can have different visualizations as you need, and they are detached from the actual data. It's a very powerful design pattern. It's like the zoom levels that were mentioned here, but a more general one. Okay. So the nice thing is I think the ideas for all that are actually implemented, and I can show you a short clip of how I think it should look like. And this is how it should look like. Unfortunately, I come from Canada, and I could only get the French version of that clip. So I had to work on the translation, so hopefully you'll be able to follow. And this is how it looks like.
Dois-je inclure les comptoirs à gaufres belges, monsieur?
Non, c'était une question rhétorique, fais-moi voir. À quoi ça ressemble selon toi, Jarvis? C'est semblable à un atome, auquel cas le noyau serait juste là. Accentue l'unisphère. Éteins les promenades. Débarrassement.
Que tentez-vous d'accomplir, monsieur?
Je découvre. Correction, je redécouvre un nouvel élément, je pense. Dégage les jardins, les arbustes, les arbres, les stationnements, les sentiers, les entrées. Agence les protons et les neutrons avec les pavillons et le jardin. Tant que t'es mort, et tu me ramènes encore à l'école.
Il me proposait de faire représenter un substitut viable au radium.
Merci, Jarvis.
Malheureusement, il est...
So my French is not so good, so there were some inaccuracies. In Iron Man 2, he actually speaks about his father, not his brother, if you remember the movie. But apart from that, I got it right, I think. Anyway, so if you think of what I just showed you, I mean, seriously, right? It's exactly about what I just talked about. So appropriate filters and control nodes. There are discrete or real, alternative representation, switch the entity focus as he moves along, a matching rendering agent. Obviously, there's a relational DB behind it, and a flexible interface definition that enables to do that, and a visual proxy design pattern. And of course, there's other things too, 2D and 3D display, seamless natural language, voice control, gesture control, and it's cool and fun, right? And actually, if you think of cool and fun, I think this is a serious comment about software that we develop. We should have them, at least the cool and fun factor, right? So you can actually relate to it, as we're too serious in a sense. OK, so I'll move to the next subject. And yeah, I still have a bit of time. Alternative splicing regulation to cover in my talk. And basically, this is how it's usually displayed in scientific papers. So this is the alternative exon, either excluded or included. And as you can see, there are many, many elements that may bind here, enhancers and repressors in the introns or the exon. There is motif, sequence logos that you probably all know that represent the splice junctions here, et cetera, et cetera. They interact. And these are kind of the illustrations that we're used to see. And the question came in a review by Burge and Wang in 2008, asking, can we move from a parts list to an integrated splicing code? So can we, they set as a long-term goal to determine a splicing code as a set of rules to predict splicing pattern directly from sequence. And so sort of to generalize what they said is, can we take everything that was derived in the, since splicing was actually found, and all these bits and pieces of information of things that we know are working for various examples, can we combine them together to look at a new axon that we've never analyzed before? Can we predict what's going to happen in a tissue-specific or a condition-specific manner? Can we quantitate how well we're doing and maybe then look more closely at the places where we fail to learn something new? Can we exploit everything that was already known to explore new things? And that's basically what we did. And that was what was introduced in that Nature paper. And just to walk you through how it works, so again, we take this alternative exon in the middle, flanking exons here, and the introns. We extract features from all these regions. Features are over 1,000 of those, known motifs, new motifs, sequence conservation, RNA structure, et cetera. And we parse them into the splicing code along with the specific tissue type that we're looking at. And the splicing code gives us predictions for the chances that we're going to see differential inclusion or exclusion for that specific exon, given its features, in that specific tissue type. That's basically how it works. But what I'm going to, and we quantitated it, and we validated, et cetera, et cetera. But what I want to talk about is, how do we actually visualize the code that we learned, right? There's a splicing code here in the middle. How do we visualize it? So we were pondering about this, and we were not sure. We eventually came up with this type of representation, OK? So what you see here is, this is the alternative exon. This is the downstream intron. So for each of those, here you see just 2 regions. And for each of the 4 tissue types that we identified, central nervous system, CNS, muscle, embryo, and digestive, and the tissue-independent one. And for each feature, this is a motif binding FOX and no splice factor. We analyze whether it's enriched in exons that are differentially included in that specific tissue. And higher the enrichment, the higher the bar. It's a red bar. And if it's in exons differentially excluded, then it's a blue bar. And then we map it to the regions. We get this, which is a very nice correspondence to what is already known about FOX in this region, and none in the A exon, in the alternative exon, which is, again, a match. But this is just a small part of how the code looks like. Then we can look at the entire splicing code that we had as a figure, and it looks something like that. So a bit intimidating, but now we have all the regions, many, many different features, and we can see their contribution, how they're enriched, and in which ones they actually show for all the different tissues. Okay, and there's a lot of stories that came out of it, and we validated some of the, so there's a lot of known stories here, but a lot of new ones that we actually went and validated. So that's how we we just didn't have any way to visualize the code. You might argue whether it's good or bad. And we didn't have a program to do that. We actually wrote it through MATLAB all the way to producing the PDFs, which I do not recommend. Okay. Yes, but yeah. But anyway, what it doesn't show you, and you might come up and say, obviously it doesn't show us, is the interaction between the different features, right? How do they tend to combine, which is a big thing in the splicing code. So we also did this, and this is straight out of using Cytoscape. So we output our algorithms into the right format, that Cytoscape, which I'm guessing a lot of people here know about. And we use this diagram with this notation. So this is color map for the different regions, the alternative exon and the flanking exons and the intron regions. Shape represents whether it's a motif or a structural feature. If it has this black edge, it's depleted a feature. And then as you could expect, the width of the edge represents the significance of the overlap, or if they don't tend to overlap. Okay. So, but these are separate representations, of course. So this is a review of our work that hasn't been published yet, but the authors kindly sent us this they basically analyzed our code and came up with this alternative representation that I'm showing you as another way to illustrate, to combine both figures that I just showed you. So these are mapping the various features for where they show up. Bigger letters means they're more important, and now you can see the interactions between them, and then blue or red, whether they're depleted or enriched, et cetera. It's interesting because it's awfully similar to what we started off with. The reason we did not go with this is, if you think of it, this is a very nice illustration if you want to review something very specific. It's very hard to write a code that will actually do all that with the layout automatically. And actually, this is only for muscle tissues. If you want to compare the different codes across tissues, it's again very, very hard. Okay. And I don't have a lot of time, so I'll skip through how we visualize the different elements and where they map in the intronic regions. But this is the enrichments of the motifs, and this is where they actually are assumed to be contributing to the splicing code. And we did a lot of mutations to verify that it's true in neuronal, non-neuronal cells with nice correspondence, and identified a lot of motifs. The idea is that you can use this analysis to direct your mutagenesis analysis, whether you're doing double and triple. And you can also plug it in. You can plug in your exon of interest, and you can have the same type of visualization within the genome browser. Okay. So very open question, though, that has to do with visualizing the regulatory mechanism that govern splicing that I'm very much interested in, is how do we represent complex combinations of regulatory features that are coming out of the code? How do we visually compare different codes, which are things that we're interested in? Say, the code for mouse versus the code of human, or maybe slight variations of the code under different tissues, et cetera. How do we visualize combined effects of motifs if I want to direct mutations to do gene therapy or whatever I want to do? How do I visualize the effect of the combined mutations, visualizing of more complex events, and of course, building a real splicing interactive and integrative tool. And I want to finish with a sort of a general comment about visualization in scientific papers. So I think right now we're here, which is very nice, but we want to be here, right? And I know, this is okay, this is sci-fi, okay, this is fine, but I think we're still not where we should be, in a sense. And so that's a good place this conference is a good place to start with. And I think if you look as an example, if you look at journals on the iPad or tablets, where you get the ads to be dynamic, right? And you compare those to what we get in Nature or Science or whatever scientific journal that you have, it's all these static 2D images. And I don't see a reason why we shouldn't be moving into interactive or dynamic visualization of scientific results within our papers or part of the scientific discussion. And For that, we need dynamic visualization. We need interactive visualization. So we need to define standards, and we need to develop tools so people can use them. And I think that requires concerted long-term effort. So it's not just one researcher applying for a grant and developing it over 2 years, maybe larger bodies and maybe involvement of major publishers in supporting this endeavor, something like that. And just to acknowledge people who helped me through this work, Ben and Brendan, who brought me originally to Toronto, and all these people in the lab that were working on the project and different aspects of it, all the funding. And thank you for your time.